The overall goal of this procedure is to isolate extracellular vesicles using a paper-based platform. This is accomplished by first laminating with two polystyrene sheets, with circular cellulose paper cutouts aligned on an array of registered through holes. The second step is to chemically conjugate molecules that would bind extracellular vesicles to the paper test zone. Next, the biological sample can be pipetted directly onto the paper test zone, and extracellular vesicles are isolated after the rinse step. The final step is to characterize captured extracellular vesicles using electron microscopy, ELISA, or transcriptomic analysis. Ultimately, subsets of extracellular vesicles from as small as 10 microliter serum samples can be purified and concentrated.
So the main advantage of this technique over existing methods like ultracentrifugation is that it's much gentler on the sample and it's simpler to perform. Demonstrating the procedure would be a grad student, Chihchen.
Starting with two sheets of polystyrene, create an array of circular cutouts, each exhibiting a diameter of less than 5 mm with either a manual hole punch or CAD automated cutter plotter. In addition, cut out a series of 5 mm diameter chromatography paper discs with a hole punch. Place one polystyrene sheet on the bench and overlay a single paper disc on top of each corresponding through hole. Sandwich the paper discs by registering and overlaying the second sheet of polystyrene. Check the overall alignment before bonding the sandwich in a thermal laminator. Next, begin chemical surface modification.